do best to go through the answer keys and explain what the heck is going on here. So, first question. Scientists studying genetics in pea plants discovered a cross of two traits that did not follow expected patterns of inheritance. Purple flowers F are dominant to red flowers, lowercase f. Long pollen grains, capital G, dominant to round. Figure 1 shows a test cross, okay, between purple flowers with long pollen grains and red flowers with round pollen grains. Okay, so here you can see the dominant traits, okay, homozygous for both, crossed with homozygous recessive, and in the first generation they were all purple flowers with long pollen. And then they allowed them to self-pollinize, okay, self-fertilize, and now you notice that we don't have our nice 3-to-1 ratio, okay? So based on the information, what is the predicted ratio of pea plants with purple flowers to the total number of flowers in the F2 generation? Well, purple flowers, 72% plus 5% is roughly 77%, so roughly 3/4. So your answer to this one was C, 3/4. Using the information in figure 1, describe the genotype and phenotype of the pollen grains. And we're supposed to drag, but we are going to write. So genotype of pollen grains from the F1 generation pea plant. Well, this is F1, and you can see that we started with homozygous dominant, homozygous recessive. So all of these guys are hybrids. Okay, so this is our big G, little g. Okay, the phenotype of the pollen grains. Oh, am I using the right letters for pollen grains? Yes. So G for the pollen grains. From the F1 generation pea plant would be, well, what do they look like? Phenotype. Can you physically see? And that would be long. Wow, this is kind of a cool feature on Word. Okay, moving on. Predict the percent of offspring with round pollen grains that would normally result from a cross between two pea plants that are heterozygous for long pollen grains. So we're using the letter G, not that it matters, but they are both heterozygous. So I'm basically taking big G, little g, and I'm crossing it with big G, little g. So if I were to set up a Punnett square, right, G, big G, little g, and I fill it in, and round is going to be the recessive, okay, because this is like two big G's, so that's going to be long, big G, little g, that one's going to be long, big G, little g, that one's going to be long, and then 1 square out of 4 will be round, okay, so that is roughly 25%. Okay, one more here. Identify, based on figure 1, so this is my figure 1, identify each genotype for flower color that is present in the offspring from each generation. So in the F1 generation, what is the flower color? Well, they were hybrid. So that was big F, little f. All of them were big F, little f. Now in the second generation, we see a mix. Okay, we see purple flowers with long pollen. Okay, so for sure, big F and big F. Okay, purple flowers with round pollen. Well, that might be the hybrid. And then we also see red. Now you might be wondering, well, Mrs. Erickson, how do you know the difference between these two? I don't. When I make a Punnett square, okay, and we are focused on F, which is flower color. Well, these guys are hybrid, so it's big F, little f. So when I allow them to self-pollinate, sorry that I can't do this faster, I see big F, okay, I see big F, little f, and I see little f, little f. So in that second generation, you will see all 3 different types of phenotypes. Wow, those Fs did not look like Fs. Okay, moving on. Figure 2, the genes for flower color and pollen grain shape in pea plants are located on the same chromosome. Pea plants have 2 copies of each chromosome. Chromosomes that carry the same genes are called homologous chromosomes. These are my homologous chromosomes, one from mom, one from dad. You can see that this is chromosome 1 from both mom and dad. And so maybe A is mom and B is dad, okay? Chromosomes that carry the same genes are called homologous chromosomes. Figure 2 shows a model homologous chromosomes with 5 genes. 1, 2, 3, 4, 5, okay? Based on the figure, which statement accurately explains how the genes on each chromosome function? All right, so let's go through these statements. Gene 1 on chromosome 1A produces the same protein as gene 2 on chromosome 1B. Right there, wrong. This gene codes for, we'll just say protein 1, and gene 2 codes for protein 2. So that statement is incorrect, okay? If this might be the gene for, if we're pea plants here, flower color, and this one might be for pollen grains, and this one might be size. All right, let's read on here. Gene 2 on chromosome 1A. So gene 2, chromosome 1A, produces the same protein as gene 2 on chromosome 1B. All right, here I have a capital E, and here I have a lowercase e. So that is false, okay, because they are not. This is the dominant form, this is the recessive form. The proteins are slightly different. Gene 3 on chromosome 1A produces a different protein than gene 3 on chromosome 1B. Well, I can see here they are both big F, so that is false, incorrect. Just make sure this last statement is right. Gene 4 on 1A produces a different protein than gene 4 on chromosome 1B. So that is true. So I'm going to circle that. Okay, moving on. Alleles that are located near each other on the same chromosomes are often inherited together. Based on figure 2, so this says near each other are often inherited together. Which alleles are least likely to be inherited? That means that they are far apart. Okay, so if I scroll back up here, my options are D and E. Okay, there's some space. Okay, that could be the answer. Let's see, G and H. Oh, that's closer, so it's definitely not G and H. D and G. D and G, wow, that is even further than D and E, so that's a pretty good candidate. And then F and G. Oh, F and G are way too close. So my answer for that one is D and G. Okay, switching gears to guppies. Guppies are small freshwater fish that are found in small streams and pools. Though female guppies are always dull-colored, male coloration varies. Many male guppies have bright coloration and ornamental spots. Figure 1 shows a basic comparison of a female guppy, a dull male guppy, and a bright male guppy. So there's my female, and then my 2 males, one's dull, one's bright. Scientists studying guppies in the wild noticed considerable differences in male coloration dependent on the presence or absence of predators. In one study, scientists placed a pair of differently colored male guppies that had similar swim-in behavior in a tank with a pair of female guppies and measured the average number of times a female guppy visited one of the male guppies in the tank. Scientists concluded the first test with predatory fish absent. Then the scientists repeated the test with predatory fish present. Graph 1 shows the scientists' results. Using the data in Graph 1, construct a statement that explains how female guppies select male guppies. All right, so if we take a look at this, it says visits by female guppies every 30 minutes. Bright male versus dull male. So when there are no predators, females prefer bright males, bright colored males. But when predators are present, they still prefer bright males, but there's not a big difference between bright males and dull males. It's pretty close to each other. So select the words that complete the sentences. So here are your word options. Female guppies prefer, well, according to the graph, bright males, okay? Outranked are dull males, regardless of the absence or presence of predators. Female guppies are, what do you guys think? Well, if we look here, this is 9, and this is like 2, 3, I don't know, 2, slightly over 2. It's not quite 3, okay? So female guppies are almost half likely to approach this type of male guppy, equally or almost 4 times. Well, that's roughly 4 times the difference between here and here. So C is the correct answer. All right, moving on. Next page. This is a new tab now. Scientists conducted additional experiments in which they looked at the effect of swimming behavior on female selection. Timid males are less likely than aggressive males to approach a predatory fish. Aggressive males would regularly swim up to a predator to make a closer observation. Scientists tested the response of female guppies to male guppies that had different combinations of coloration and swimming behavior. Graph 2 shows the results when predators were present. So here we have number of female guppies selected males, timid males, this would be bright and dull, and then our aggressive males. Graph 3 shows the results when predators are absent. Okay, so this is in the presence of predators and here absent. So we can kind of see almost a flip with our bright males and our dull males. Okay, construct a statement that explains the data in graph 2. Select the words that complete the sentence. In the presence of predators, okay, over here, blank male guppies that exhibit blank swim behavior have the greatest reproductive advantage. Well, if we want the higher bar graphs here, so the answer would be dull. Do I still have my pen? Yes. Dull, because blue beat red every single time. And what kind of behavior were they showing here? Which one's higher, aggressive or timid? And it would be timid. Which statement best predicts how the presence of predators will affect a population of guppies over time? The number of dull male guppies that exhibit timid swim-in behavior will increase the population. Well, that's a true statement. The number of bright male guppies that exhibit timid swim-in behavior will increase. I guess you could argue for that, but our timid dull males outnumber, so A still holds. The number of dull male guppies that exhibit aggressive swim-in behavior will increase. Well, those numbers were lower, so it's definitely timid behavior here. And the number of bright male guppies that exhibit aggressive swimming behavior will increase. No, not so much. So A is the best option on that one. Okay, scrolling. Okay, tab C. The scientists conducted an experiment in which predatory fish were presented with a pair of male guppies, one dull, one bright, and recorded how many predatory fish approached each guppy. The scientists determined that the percentage of predatory fish that approached the bright male guppy and the percentage that approached the dull male guppy. Table 1 shows the results. So here we have bright. Predators prefer bright male guppies because they stand out versus your dull because they are more camouflaged, right? So scientists claim that the change in behavior of female guppies in the presence of predators is a beneficial trait. What additional evidence will support the scientists' claim? All right, data showing that dull, timid males are least likely to be eaten by predators. Yeah, if you're dull, you're going to blend in. If you're timid, we saw that they were selected more, so that's a true statement. I'm going to circle it and come back. Data showing that bright, timid males are least likely to be eaten. No, they were brightly colored and approached more. Data showing that dull, aggressive males are least likely to be eaten. No, because those numbers are lower, so that's not true. Data showing that bright, aggressive males are least likely to be eaten by predator. No, that's not true either. So A, best option. Okay, based on the information in Table 1, here's Table 1. Which statement describes the effect predators most likely have on the population of guppies? Let's read through them. As the number of predators increases, the percentage of dull male guppies in the population will increases. Okay. That makes sense because these numbers imply that they were eaten, okay? So if you increase predators, you're going to see more dull males. So that's true. As the number of predators increases, the percentage of bright male guppies, no, that's false. They will get picked off. As the number of predators increase, the percentage of dull female guppies in the population increases. So this has nothing to do with females, so can't be the answer. As the number of predators increases, the percentage of bright female guppies. And again, we are not looking at females, we are looking at males, so A. Scientist observes 2 populations of guppies. The scientist's observations are recorded in the guppy population table. So we have X and Z. Bright male guppies, 90% of the population versus 20. Wow, that doesn't equal 100. Wait, oh, it equals 100 across. Oh, okay. Whew. So 90% bright, 10% dull, and then 20% bright and 80% dull. Based on the evidence, which statement most likely explains the scientist's observations? Okay, so here we see bright male guppies. So do we see more predators? No. Okay, so basically these guys are no predators. So I'm just going to put no P in here. There are predators because there's lower amounts of them. So population W has more predators than population Z. Nope. Population Z has more predators than population W. Yes. Population W has more female. We are not looking at female guppies, so we can just eliminate those two. So B is your answer. Okay, this question kind of correlates with this. Use the evidence provided in tabs A through C, basically everything we've read so far. Identify the selective force acting on each population. Select the correct answer for each row. So W, do we see predator selection, okay, or mate selection? And so with W, because we have bright male guppies, females prefer bright male guppies. So this is a mate selection, female choice. Whereas with Z, we have predators, okay? And so it's predators that are deciding which types of males we see. So that is what that question was asking. Okay, moving on to sex determination. Sea turtles, there are animals whose sex are determined by temperature at which the eggs incubate or develop. Sea turtles are one of those animals. Graph 1 shows the percentage of female sea turtle offsprings for different average incubation periods. Wow, I can't read. Temperatures. So female offspring percentage, average temp. Okay, the period when sex is determined is known as the thermosensitive period. Thermosensitive period in sea turtles occurs in two phases that overlap slightly. During the first phase, genes that control female development are turned on or off, dependent on the average incubation temperature. During the second phase, genes that control male development are turned on or off, dependent on average incubation temperature. Figure 1 shows the effect of incubation temperature on sea turtle development. So here's that. You can see it goes from 0 to 60. And then this is our thermosensitive period. And then dots represent female producing temperature and males are these lines, and there's that overlappage. Okay, based on the data in graph 1, boom, graph 1, what is the average incubation temperature at which 50% of sea turtle offspring are female? So you just find 50% and you just like move your line over until like the line intersects. And so I did a bad job drawing my line, but it's roughly 30 degrees Celsius. Okay, down here. Based on the information in graph 1 and figure 1, construct a question scientists could ask to determine how environmental conditions and time affect the development of female sea turtles. So which genes are turned on or inherited? Well, are we talking about inheritance here? No. So turned on is the best option there. Of development when eggs are exposed to a temperature that is above 30 degrees Celsius. Oh, I missed a box. Which genes are turned on during days? Okay, so then which days determine determine a female. So we're focusing on female turtles. So which days are critical for female sea turtles? So in our information that we read, here's our female producing temperature. And it's going to be between 15 and 30. So B. All right, a sea turtle lays 100 eggs that are incubated at an average temperature of 29 degrees. Using the data in graph 1, predict the number of sea turtles. So we just find 29. So that's like 30. I'm just going to nope, I'm using my graph wrong. My bad. Here's 29 degrees Celsius. I'm just going to move up here. And there's my answer. It's going to be 40. I'll have roughly 40% female sea turtles. And out of 100, it's 40. Yeah. Okay, so what's 40% of 100? 40. Okay, tab B on the sex determination of sea turtles. Scientists studied 4 genes that control the development of male and female turtles. They measured the gene expression ratio of these 4 genes during thermal sensitive pyramid. Graph 2 shows their results. So gene expression ratio during the thermal sensitive pyramids. We have gene 1, gene 2, male, gene 3, male, gene 4, back to female. So genes 1 and 4 were higher in females or female producing temperatures, and genes 2 and 3, male. just going to continue to read. Nope, I think I'm going to answer this question. Using the data in graph 1. So this is graph 1. And graph 2. This is graph 2. To construct a claim about the role of gene 4. So we're going to focus on gene 4. In the sea turtle development. Gene 4 is most likely to be expressed during the thermal sensitive pyramid when the temperature is what? Okay, so we're going to use this. And our options are above 30 degrees or below. Well, we see more females because this is our halfway mark. Above 30 degrees Celsius. So 30 degrees Celsius, anything above that is going to be female. So we want above. Okay, so that goes there. This indicates that gene 4 is most likely involved in developing female traits. And I got that because this bar graph, orange is higher than blue, purple. Based on the gene expression ratios shown in graph 2, determine which genes mainly control female development and which genes mainly control male development. So I think my table got cut off. So I just need you to circle which ones are female. And so we see the highest orange in gene 1 and gene 4. So you would shade in gene 1 and gene 4. And if there was a column over here for male, then then it would be, gene 2 and gene 3. Okay, graph 3 shows the predicted global temperature change in degrees Celsius under 3 conditions. No change in fossil fuel use, which is blue, some reduction in fossil fuel use, which is red, and rapid reduction in resource usage. Okay, and then we have a range. Which claim about the effect of the predicted temperature change in graph 3 is supported by the data in graph 1 and figure 1? Okay, so if we come back up to here, okay, I said anything above 30 degrees Celsius is like going to be female dominant. And so we are seeing a temperature change of all of these showing an increase. So the trend is the percentage of female sea turtles will increase. It doesn't talk about number of eggs laid. Males, it needs to be colder. And that also, we're not reading anything about that, okay? And then this is a question that you don't need to answer. Just know that you may be asked because you might have to use the information that you are presented to answer and predict what might happen to sea turtle development worldwide. Okay, moving on to malaria and mosquitoes. We're getting there. Malaria is a disease caused by the parasite Plasmodium, which is a single-celled protist. Malaria can be transferred to humans through female mosquito bites. Female mosquitoes use blood to develop their eggs and can transmit Plasmodium between animals as the mosquitoes feed. Plasmodium affects humans in stages. Figure 1 shows the life cycle of Plasmodium within a human host. So there's the mosquito, and it gets in, and then it finds the liver, and it looks like it gets inside some liver cells, and makes more Plasmodium, and they bust out and they just continue to infect red blood cells and spread. And then mosquito bites human, it consumes the Plasmodium, and then it can sexually reproduce in the mosquito. Okay, so there's the steps underneath it. Using the information in tab A, construct a statement that describes the activity of Plasmodium cells in the liver. Plasmodium cells reproduce in the liver using what? So here it says reproduces asexually. Keyword right there, asexually, one parent. The only way they can do that is by making a copy of themselves. Meiosis is the production of sex cells. Okay, that is sexual reproduction. So if you are doing something asexually, it is mitosis. Okay, in order to blank, increase in number or introduce genetic variation. Again, genetic variation is tied to sexual reproduction. So increase in number is our answer. So A and A. Using information, describe one way that genetic variation increases during the life cycle of Plasmodium. During its life cycle, Plasmodium increased genetic variation when? All right. So we're going to continue to read through and then look at our options. Fertilization or mitosis. Well, which one would help them increase genetic variation? And that is tied to sexual reproduction, so fertilization. I'd have to get back into the mosquito to reproduce sexually. All right, and it occurs in, where does it occur in? Well, reproduces sexually in the mosquito, and so C is your best option. Scientists conducted studies to determine if mosquitoes infected with Plasmodium were more likely to land on a surface coated in a human odor. Scientists also compared the land-in rate of mosquitoes infected with Plasmodium with the land-in rate of unaffected mosquitoes when the human odor was absent. Their data are shown in graph 1. So, no human odor. Okay, so the human odor is absent. Plasmodium-infected mosquitoes kind of low. Human odor present. They are very high in comparison to unaffected mosquitoes. Another group of scientists studied the preference of mosquitoes for organisms with Plasmodium and organisms without Plasmodium. Their data are shown in graph 2. So organisms with Plasmodium, preference of mosquitoes, higher land-in than those that did not have the Plasmodium. Okay, multiple choice. Which statement best explains the data in graph 1? Okay, let's read through the statements. Mosquito populations are negatively affected by the presence of the Plasmodium. Graph 1, tab B. Mosquito populations are negatively affected by the presence of Plasmodium. I'm not seeing anything about that. So, because this is looking at human odor. Plasmodium populations are negatively affected by the presence of mosquitoes. No, they are positively affected. They need mosquitoes in their life cycle. Plasmodium populations benefit by affecting the behavior of mosquito populations. It could be plausible. Mosquito populations benefit by affecting the behavior of Plasmodium populations. I'm not seeing that either. So C. It's got to be our best option there. Okay, we got a drop-down menu, fill in the blank. Explain how the data in graph 2 support the claim that the behavior of mosquitoes benefits the Plasmodium population. The likelihood of mosquitoes landing on organisms infected with Plasmodium is, all right, so with Plasmodium, without, you can see that it is going to be greater because it's higher than the likelihood of mosquitoes landing on organisms that are not affected. This blank, the likelihood that the Plasmodium will survive. Increases, decreases, or has no effect. Well, it's going to be increases because then they can reproduce sexually. Okay, switching gears to Caribbean anoles. Caribbean islands, shown in this map, are home to about 150 species of anoles, these guys over here. Though each island is surrounded by water, there are 4 distinct types of anoles that are found on every island. Caribbean anoles live in the diverse habitats and vary greatly in size and other physical characteristics such as leg length, skin color, and toe pad size. Okay, and then toe pads provide friction needed to walk across smooth, flat surfaces. Species of the Caribbean anoles can be categorized into types according to their body characteristics and the ecological niches they occupy. Figure 1 shows the 4 types of them and some characteristics. Okay, and then the habitat. Okay, so the twig anole is the only type of anole that lives on the narrowest of branches of trees and shrubs, based on the data in Table 1. So this is Table 1. Select 3 statements that could explain how a characteristic is an adaptation for moving across thin branches. So adaptations are traits that give them a benefit in their environment. Okay, so we're looking for things that will help them walk across the narrowest of branches. All right, let's read through. So we're going to focus on the twig. No, this column right here. Okay, large bodies. Do they have large bodies? Kind of intermediate. Have more balance than small bodies and narrow twigs. I don't think that's true. Oh, here we go. Habitat, canopy of trees. Oh, this is like cut underneath. Okay. Short tails are better than long tails for walking across narrow twigs. So they're short and they do that, so that's a check. Gray bodies are more easily seen than green bodies on narrow twigs. Gray bodies are more easily seen than green bodies on narrow twigs. Okay, these are them. I'll come back to that. Short limbs are better than long limbs for walking across narrow twigs. Yep. So basically we're just checking off things about their characteristics so far. Small toe pads provide better suction than large toe pads on narrow oh my god, I can't read. Twigs. And that is true. Now I'm going to come back to here. Yes, they have gray bodies, but that has nothing to do with them walking across narrow, thin branches. So those are the three we're going to pick. Okay, the trunk crown anole spends more time than any other anole walking on large, flat leaves. Based on information in Figure 1, okay, so we're going to focus on the trunk, this guy. In Table 1, so the next column over, which characteristic of the trunk crown anole explains how it adapted to walking on large, flat leaves? Green color? No, they're not green. Long tail length. Okay, they do have a long tail. We'll come back. Medium body length. Body length. Oh, I'd say that's medium. We'll come back. Very large toe pad size. Large flat leaves. Huh. Am I looking at the right one? Yes. Oh my god, no I'm not. Oh my god, ugh, I'm looking at the wrong column. Good lord, I'm supposed to be looking at this one. I'm going to change colors. I'm so sorry. I'll do this really cool blue one. I'll do this again. Are they green? Ugh, yeah, they're green. Oh my gosh. Okay, so does it help them walk on large flat leaves? Maybe we'll come back. Long tail length. Medium, so it's not that one. Medium body length. Yeah, it's kind of medium. Very large toe pad. Very large. Okay, but now adapted to walking on large flat leaves. Which one of these characteristics will help them do that? And that is going to be D. I'm so sorry. Just wasted like 3 minutes on that. Okay, moving on to stickleback fish. After an earthquake in 1964, freshwater ponds formed on multiple islands in the Gulf of Alaska. Some ocean fish were trapped in these freshwater ponds. One of these fish, the marine stickleback, was able to adapt to the freshwater conditions. Stickleback fish have different kinds of armor. The marine stickleback fish has complete armor with many bony plates on its sides and back and a pelvic spine on its bottom side. The freshwater stickleback fish has low armor with fewer and smaller bony plates and a smaller or even absent pelvic spine. Figure 1, right here, compares the general structures of marine and freshwater sticklebacks. Here's my marine and here's my freshwater. Based on this information, determine if each structure of a stickleback is an adaptation for ocean or freshwater. Pelvic spine. So here we see a pelvic spine on the marine, but we don't on the freshwater. So is it ocean or freshwater? We would circle ocean. Low armor. Wow. Looks like these guys have more armor than the freshwater. So, but it says low armor. So these guys have more, so these guys have low. So that would be freshwater. Tricky, tricky. Complete armor. And that would obviously be our ocean. They are armored. Okay. In stickleback, the presence of a pelvic spine is dominant to the absence of a pelvic spine. Predict the percentage of offspring with a pelvic spine that would be produced from a cross between a stickleback fish that is homozygous recessive. 2 little p's. I'm going to make a Punnett square. P, these are my little p's. And a stickleback fish that is heterozygous. Big letter, small letter. So this is a big P and then a little p. So when I fill this in, I get big P, little p, big P, little p. And then the bottom ones here are going to be 2 little p's across. I wish I could find a way to copy this faster. There. What percentage of offspring will have a pelvic spine which is dominant? You would write down 50. 2 out of 4 boxes have a big P in them. Stickleback fish is dominant. Okay, predict a percentage of offspring with and without a pelvic spine that would be produced between fish that are heterozygous. So that's a big P, little p. Okay, so one parent is big P, little p. Second parent is also big P, little p. Okay, this will give me a 3-to-1 ratio. 2 big P's, big P, little p, big P, little p, and then 2 little p's. So pelvic spine, 75%, and then without would be 25. Oh my gosh, we're gonna make it. Final page. Woo-hoo. Scientists study a population of stickleback fish in Loughburg Lake in Alaska. In 1988, the population of freshwater stickleback fish in the lake died off. By 1990, a small population of marine stickleback fish moved into Loughburg Lake. Okay, so they were gone, and then freshwater died off, and then marine came in. Okay, graph 1 shows the percentage of stickleback fish with complete armor and low armor in Loughburg Lake. So remember, complete armor was our marine, and the low armor were the freshwater ones. Okay, so low armor, freshwater possibly, complete armor goes down. Okay, let's just move on here. Select the words that complete the sentences. The percentage of stickleback fish with complete armor, that's white, and you can see it decreased. In Loughburg Lake, during this time, the number of predators in the lake most likely, well, why would they have complete armor? Maybe there wasn't a lot of predators, so also decreased. Stickleback fish with complete armor also have a pelvic spine. During which year was the percentage of stickleback fish with a pelvic spine most likely lowest? In Loughburg Lake. Okay, so usually stickleback fish that have armor have the pelvic spine, and so we don't really need the pelvic spine, so following the complete armor must be most recent, 2008. Final question. Which statement best explains the pattern of armor in stickleback fish shown in graph 1? Complete armor is dominant to low armor in fish. That is false. Decreases, and doesn't really say if it's dominant or recessive. Low armor provides less protection than complete armor against disease. Nowhere does it say that. Stickleback fish with complete armor are better able to avoid predators than stickleback fish with low armor. Maybe. Stickleback fish with low armor are better able to survive and reproduce than stickleback fish with complete armor. Maybe. So if our predators are decreasing and the low armor is going up, and nowhere are we really talking about predators. So going back to this question, I mean, yeah, it's like 50-50, but we're not really talking about predators. When you see things that are becoming more common, that has to do with passing on traits, kind of like my funny clownfish story. There will be more funny fish. And so I think D is going to be your best option there. And I can see why this packet was difficult. Wow, even threw me for a loop. So there, made it through. Why don't you just write down at the top how many you got right, because I am super curious. There were 32 questions. We'll do some more MCA review tomorrow. But all right, bye.